My name is Cullen Buie. I'm an associate professor of mechanical engineering at the Massachusetts Institute of Technology. I'm also the co-founder of a company called Kytopen, which is looking to accelerate the delivery and commercialization of cellular therapies. I have a laboratory where what we're doing is using microfluidics, which is fluid flow in very small confined areas. So we flow fluid in channels that are roughly the dimension of a human hair, so the width of a human hair. At those scales, you can run a lot of interesting experiments on biology. And so my group uses those very small channels to do experiments on microbiology, such as we run experiments to deliver DNA and gene-altering material into bacteria to change the way they function. And we also run experiments to understand how different bacteria in communities communicate with one another. Almost think of it like social networking for bacteria. We want to understand how bacteria communicate in the world and how that communication and those interactions affect human health and other applications. So we develop tools, you might say, that help biologists and applied biologists do the type of work that they wish to do. So there are those who are interested in, for example, things like the human microbiome. So the microbiome is all the bacteria and microorganisms that live in and on your body. And this is a new and exciting area of research, but we're very much limited in the tools that allow us to probe those spaces. So in your gut, for example, there are thousands of bacterial species that live there. And right now, it's very difficult to understand how they interact with one another. So the tools my group is presenting and developing helps those that are interested in understanding fundamentally or in an applied manner how those interactions are occurring. So it's very much engineering. It's engineering to solve a problem that enables biology. I've always been curiosity driven. When I finished my PhD was around proton exchange membrane fuel cells. So these are fuel cells that use energy. They take energy from a fuel such as hydrogen and they convert it electrochemically to give you useful energy, waste heat, and water. And in my postdoctoral study after my PhD, I started looking at microbial fuel cells. So microbial fuel cells do that exact same process, except they use living organisms, bacteria, in order to catalyze or initiate those reactions. And so from that work as a postdoc, I started drifting towards applications that had more of a biological focus. Then in 2010, a lot of tragedy struck with my sister who passed away. My sister she died of sepsis, which is a bacterial infection that goes awry in your body. And after that, I started to become more curious around just bacteria in general, the things they do. It was shocking to me that in 2010, a doctor could die of a bacterial infection. That seemed kind of ridiculous. My sister was a physician. And so that kind of further pushed me towards this space. I'd say another piece of ingredient was really being in Boston. So Boston is really a biotech and biotech hub, a hub for applied biology. And here you are within a 5 or 10 minute drive or Uber from world exports in all kinds of areas. So as I started becoming interested in sepsis and in bacterial infections and in microbiology, there were all kinds of people that I could interact with at Harvard and MIT and Tufts and Northeastern to help me understand some of these problem spaces. And so my work started drifting towards applications that could be used to understand the microbial world. So the question is a good one. The question is, it seems that at one point I was going the academic route, and then now my work seems to be going towards things that are more applied. And where do my interests lie? Do my interests lie more in the academic space or in the industrial space? And I would say the answers are both. I'm interested in taking the understanding that we were able to gain from my laboratory and translating it all the way out into the world. I tend not to be a planner. So when I got into academia, I didn't plan to be in academia for my whole life. And even now that I have a company that started up, that spun out of my lab, I'm not necessarily planning to do that my whole life. I'm doing the things that seem like what God wants me to do today. That's really all that I can say is it seems like the best way to promote human flourishing really is to take the technology that we've developed and take it out of the lab and translate it so that it can affect thousands or perhaps even millions of people. And I don't know how long that's going to take. That may take 5 years. That might take 15 or 25 years. but I want to see it through. And that's really been my perspective on things throughout. I don't actually view myself as a professor or as an entrepreneur. I'm Cullen, and I actually don't like being put into those boxes. Like a lot of people around you, they see you, well, you're in academia, so of course you're a professor, you have tenure. Why would you leave? But I only have tenure because of God's grace. And so why would I hold on to that like tenure or being a professor like it's an idol? So my perspective, and that doesn't mean that I just disregard it either. There's a lot of value in the work that I've done to get where I am. It's just that I'm trying to do what God has for me for the next phase. And I don't know what that means long term. I don't know if that means I'm leaving academia. Probably not. I don't know if that means I'm going to be an entrepreneur for my life. Probably not that either. It'll probably be some mix of both. I think it's going to be hard to come up with the exact label for what it is that I'm doing. But you see more and more academics and faculty kind of straddling these two spaces of not just being concerned with their graduate students and their teaching and their journal articles, but also being concerned with how that work is being translated out into the world. And so I'm straddling those things, and for now, that's a space that's a bit murky and it's kind of hard to define, but I like where I'm sitting.
You're like a true Renaissance man.
I don't know about that. I'm just trying to be what God has made me. And that's something that I think about often is early. So I'll give some examples. Early on when I got to academia, it's easy to look out and see other people that are successful in academia and say, I want to be like them and I want to be like them. And I heard someone give this example, and it really changed my thinking a lot. When you're in academia, you were hired in order to be yourself. So it's weird. You get this PhD, you do this unique piece of work, you get hired in academia to be yourself, but then you look out at the world and those around you and say, okay, maybe that's the person I should be like. But if you're going to be like someone else, you can only ever be second best. And what I realize is the best thing for academia and really for the world is for me to be myself. It's for me to be the person God made me. And the person God made me is not confined to academia. That's just the fact that I've seen. And so would actually be denying the world if I were to only confine myself to academia and put myself in that box and say, this is the only thing that I can do. Well, I know who I am because of Christ. I know who I am because of the Bible. I know that I am tremendously loved and tremendously flawed at the same time. I have gifts and I've also been given opportunities. I've worked hard and I've been fortunate. There are all these dichotomies that exist all at the same time, and I'm comfortable with those dichotomies. I'm comfortable with being a sinner and loved all at once. And it really takes a lot of the pressure off, knowing that Christ loves me not because of my job, not because I have a PhD, not because I'm at MIT, not because I have tenure, not because I'm smart, not because of anything that I've ever done. I know that I'm loved because of who Christ is and my faith in him. And that actually gives me the confidence and strength to go out and take risks, because let's say my company fails. Let's say I fail as an academic. It doesn't change my salvation. So I can actually take risk, because what am I risking? Everyone around us is really working to build up a name for themselves and to become something and to present something to the world that will validate them. But in Christ, I'm already validated. So I can just do the work, not because I'm trying to validate myself, but because I want the world to be a better place. And I think that actually leads to better work. I think it leads to better science on the academic side and better companies and better translation, hopefully, on the entrepreneurial side. Just in general, being in academia, you see a lot of wonderful and beautiful science and engineering. And it reinforces to me, one, that this world was created, that this world is beautiful, and that there is an order to it. The whole purpose of being a scientist or an engineer is to observe the order of the world, understand it, and then use it to promote well-being for humanity. And so I've actually become more a bigger believer in the fact that this world is created and this world is beautiful from seeing all the wonderful discoveries that have been made in the scientific and engineering world during my time in academia. So Cullen is really trying to enable the field of cellular therapies. So this might sound like science fiction, but it isn't. Today, there are therapies where they will take your blood, they will isolate certain immune cells from your blood, they will change the genetic code of those cells so that they can find your cancer, then put them back in your body and they cure your cancer. It's like a software update for your immune system. The challenge with these technologies today is that they're tremendously expensive. Just to deliver the therapies, it costs around $500,000. That's a huge economic burden on the people with these diseases and on society. Cullen actually is developing a way of uploading that foreign DNA into the cell that changes its function. That's thousands of times faster than what is done today. And we do this using automation, so we can use automation and robots in order to rapidly discover the conditions that allow you to perform this software update for these immune cells. But we also have a technology that can be used for manufacturing of these systems. Let's take one example. In CAR T therapies, you need around 1 million cells per kilogram of the patient that's going to be treated. So for someone my size, you need on the order of 100 million cells. Our technology can do 1 billion cells in 30 seconds. So doing that 100 million cells is trivial. But to put that in further perspective, the current technology that's used for this uses viruses in order to change the DNA, and that takes days. So we're taking something that's currently days, and we're going to do it in less than a minute. Yeah, so the current process for treatment in these therapies, they're called adoptive cell therapies. In these therapies, the current process is you go into a clinic or your hospital, they harvest your blood, they then take that sample, they ship it somewhere, they perform a complicated process, typically using viruses to edit those cells. And about 30 days later, that sample is shipped back to the patient and infused, and that is the drug. It's a living drug that consists of their own cells that have been edited in order to find their cancer. But in our case, because our technology can edit the cells without using viruses and can do so extremely quickly, in the future we could enable a scenario where this process takes 1 to 3 days or perhaps even the same day, where the patient could come into the clinic and leave out the very same day with their edited cells actually put back into their body. That's the vision that we're looking to enable. This can't be done with the current technology that utilizes viruses. And so in the short term, our technology will reduce the cost, but down the line, it should also reduce the burden, the clinical burden on the patient that has to wait for 30 days. And this is significant because these are people that in today's therapies have cancer and they're dying of cancer. And so 30 days is a long time to wait when you're dying. And so we want to reduce that significantly. The thing about cancer is cancer hides from your immune system. So cancer cells are cells that are simply behaving poorly. And your healthy immune system is finding those cells and eradicating them all the time. What happens in cancer is you get genetic mutations that effectively allow those cells to hide from your immune system or trick your immune system into even helping them. And so these therapies are giving your immune system a boost against the modifications that the cancerous cells have made. It's helping you find those cells that have been tricking your immune system in the past. So your immune system actually does this all the time. It's just that in cancer, you have cells that have been able to evade the immune response. There is a lot of work in this field of the microbiome, and there's a lot of excitement around the microbiome as we understand and realize how important the microbes that live in, on, and around our body, how important they are for health. And I think, though, at the same time, there's a severe lack of tools that allow us to probe this system and understand what's really happening. Most of the work we have around the microbiome, it's really empirical. You see a correlation between some disease and then with some set of microbes, but I'm healthy, you might be healthy, and our microbiomes could be very, very different. And it's extremely difficult to understand why that is. There's interactions between the microbes that are living in and around us. There are interactions between the microbes and us. And we are really only scratching the surface of the complexity of the interactions between the microbes and one another and us. And I'm actually really excited about the work going on in my lab to try to understand the communication between microbe to microbe and ultimately down the line microbe to the host, because that's really what we need to understand, is not just who is there, but who is there, what are they doing, and how are we interacting with one another. And this is a space that some are working in, but most have jumped beyond that level of understanding, and they're trying to get the therapeutics. And I think the field might be moving a little too fast. I think there's a huge need for fundamental tools that allow us to explore and understand a bit better what's actually happening in these systems. So I've been trying to come up with a better answer for the question, what do you do? So people ask you, what do you do? And I think in our society, when they're asking, what do you do, they're asking more than your work. They're really trying to figure out who you are. In our society, we define who you are by your work, and I think that's wrong, to be clear. But the answer that I'm starting to give is I use science and engineering to promote human flourishing. In the academic sense, how do I do it? I do that through teaching and mentoring and research. So when I take young students and I teach them about thermodynamics so that they understand how the world works in this particular sense, how energy is transferred, they can then use that knowledge to then engineer systems that use that understanding. So I am promoting human flourishing through my teaching. When I am mentoring a graduate student and helping them through a PhD, I am training a young mind to be able to assess a problem, determine the state of the art, and then advance the state of the art. And so I am replicating that knowledge or that information and then putting it into, or helping a young person cultivate that in themselves so that they can do that for the next 40 years. On the case of my company, we are trying to take this technology that makes these very promising therapies faster and more affordable. And by doing that, that should help people, millions of people around the world suffering from cancer, rare disorders, and genetic diseases, perhaps receive relief. Jesus was a healer, and we are effectively, through this company, trying to promote healing for the sake of human flourishing. What's the common thread? The common thread in each case, we use science and engineering to promote human flourishing. In some cases, it's intellectual and it's mentoring. In some cases, it's physical flourishing. But the common thread is the science and engineering for the benefit of mankind.